Here in California, we're lucky enough to have our own mussel species, Middleus californianus, a large mussel that you can still find mostly on hard substrates, especially in areas that are more exposed to the open seas, like on pier pilings and rocky escarpments facing the open ocean. In more sheltered areas, such as bays, the mussels you find are going to be in the Middleus edulis complex, the edible or bay mussels that are typically grown in aquaculture for the consumer seafood market. These are the mussels we'll be observing in this video, collected from the docks in Oceanside Harbor. Whether we're talking about Middleus edulis or Californianus, or clams or oysters, there will be commonalities because of their bivalve heritage. All are filter feeders. Their food consists of small particles of organic material, bits of algae, phytoplankton, detritus, etc. Things that circulate into the mantle cavity where they're collected and eaten and then converted into bivalve biomass. They are able to close their shells tightly using a fairly massive adductor muscle. That much is the same for nearly all adult bivalves. The difference between mussels and clams and oysters are largely due to where they live and how they get around, or not. Clams are expert diggers. They use their hatchet feet. Remember the other name for bivalves? Pelecypods? To bury themselves in soft bottoms. But in order for them to feed while buried, they also need to use siphons to pump clean seawater into the mantle cavity from the outside and then back out. Oysters, on the other hand, cement themselves onto a hard surface and remain there as sessile organisms. In order to get seawater into the mantle cavity, they just need to relax the adductor muscle a bit and use a bit of abduction, spreading, assisted by a different set of muscles, to open their shells, allowing the movement of seawater to provide a continual flow of food into the mantle cavity. They have no need for a muscular hatchet foot or a siphon because there's no self-bearing under soft sediments. Mussels, like oysters, also live on hard substrates and don't bury themselves, and they allow seawater to flow in and out of the mantle cavity by opening the space between the shells. They don't, however, cement their shells to the hard substrate, but rather use their byssus, a fibrous beard, that's literally the meaning of byssus, to attach to the substrate. At the other end of the byssal fibers is the hatchet foot, which remains inside the shell. Mussels are only semi-sessile. You can pull them off their substrate and place them elsewhere, and they will re-anchor themselves with their byssal threads. In order to open the mussel, we need to sever the adductor muscle, the thing that keeps the animal shut. By cutting the adductor, we can gently open the mussel, leaving the rest of its anatomy intact, and we could take a behind-the-scenes look at what goes on inside the mantle cavity of a live feeding mussel. Anyone with experience shucking oysters, clams, and mussels will tell you that the trick to opening bivalves is to know where to cut. We're always going for the adductor. If you run your blade anywhere else, you're just slicing up the animal and doing nothing to make the shells separate. In a mussel, the adductor muscle is on the dorsal side, opposite of where the byssus comes out. Now, despite this location, the only place you can insert the blade of your knife is near the byssus, and from there you need to work around to where you can actually cut the adductor. The shells will start to separate, and once the adductor is completely severed, you'll be able to gently pry open the valves to approximately a 90-degree angle and prop the shells open with a wooden stick. Then you'll want to submerge the animal completely underwater in a bowl to make your observations. First, locate the foot and the mantle. The foot is easy enough to find. It's the thing that the byssus is still attached to. The mantle is basically all the soft parts of the mussel attaching to the shell. The edge of the mantle in a mussel is frilly and dark colored. You can also see the cut surfaces of the adductor muscle. It's white. You won't see the abductor musculature because it's behind the mantle near the hinge. We're going to make observations on the gills, or ctenidia, which look like large folded leaves inside the shells on either side of the foot. That's in the space between the foot and the mantle. We've seen in the squid how the gills operate as respiratory organs. The primary function of gills in our mussel is for feeding. They provide a large surface area on which suspended food particles will make contact and get swept by cilia up to the edge of the gill where there's a groove carrying a string of mucus. The particles of food get trapped by the mucus, which is constantly being swept by the cilia up towards the mouth. We call that groove and its sticky mucus the animal's mucociliary tract, and this is the bivalve's exclusive mode of acquiring nutrients. In our normal lab exercise, we demonstrate this by putting a couple of drops of seawater containing a visible red carmine dust. You can actually see the particles of red dust moving up the sides of the gills and into the mucus thread, which moves down the grooves, where it will eventually be swept into the mouth along with all the little bits of food that the mucus has picked up. In lecture, we will have talked about filter feeding as it happens in sponges. Choanocytes trap particles at their collars and ingest them via phagocytosis. Now, what we're looking at here is a completely different mechanism of filter feeding. This time, it involves a mucus trap. Now, in a weird sort of way, you are actually doing a similar kind of filter feeding on airborne particles every time you breathe. The lining of your respiratory tract is covered by a layer of mucus that traps airborne dreck, including bacteria, fungal spores, viruses, particulate matter, etc. And then the cilia sweep the mucus upward toward your epiglottis, where the mucus is dumped into your esophagus. You then swallow the mucus along with all the little bits of food that's gotten picked up, and then you digest them. You and your mussel are not so different after all. Now, to recap the mussel anatomy, there's the mantle and the foot, and the mantle cavity, of course, the byssus, the adductor muscle, the ctenidia or gills, and the mucociliary tract. Now, in an in-person lab setting, we'd likely be taking note of some of the other things that get brought in together with those mussels. It's part of the same fouling community. This is a rather harsh name applying to all the invertebrates that encrust buoys and boats and docks. We'd likely be seeing crustaceans like little crabs, and amphipods, and isopods, tunicates, that is, sea squirts, and annelids of various sorts, brittle stars, flatworms, bryozoans, basically a large diversity of invertebrate taxa that deserves its own 3-hour lab to survey. We never have time to do a proper job of identifying and discussing them as part of the official lab activity, but there's always plenty of sharing of this off-target animal diversity. It's kind of sad to have to miss out on this aspect of the experience.